So this section is for section 9.3, and this section really details how the cell cycle is controlled and regulated. The different types of cells, especially for eukaryotic cells, because most of those organisms are multicellular, they have different types of cells, and those cells are able to divide at different rates depending on the type of cell. And the reason why those cells divide at different rates is because of different cell signaling mechanisms that occur at the molecular level, some of which we've discussed already. And then the final part of this section will kind of explain what happens when the cell cycle goes out of control, which is usually resulting in cancer. So essentially, we've discovered that the cell cycle is controlled by a lot of different specific cell signaling molecules present in the cytoplasm. And we did a lot of different experiments with mammalian cells to determine what exactly controlled the cell cycle. But in the end, we determined that there were molecules present in the cytoplasm, and those molecules helped control whether the cell was going to progress to the S phase and to the M phase. So we call this system the cell cycle control system, and this is kind of like a washing machine timing device, because there's a lot of steps that have to be fulfilled along the way. If a particular step isn't fulfilled, the cycle won't proceed. And so though some of the signals and checkpoints are internal, and some of them are regulated externally by things outside of the cell. But regardless, the cell has these specific checkpoints, and the cell cycle will stop at those points and wait until they're told to go and proceed. So they're kind of like a stop and go signal, so to speak. This is a visual representation of the cell cycle. So we've talked about the G1, the S, the G2, and the M phases. M, you might recall, is where mitosis is happening. So animal cells mostly have stop checkpoints, which are locations that halt the cell cycle until they're overridden by the go-ahead signals. And at these checkpoints, basically, there are different mechanisms involved to ensure that everything that's needed to happen has happened correctly up to that point. And in the typical animal cell, there's 3 checkpoints, one at G1, one at G2, and one at M. So for most of the eukaryotic cells, the G1 checkpoint is known as the restriction checkpoint, and it appears to be the most important because typically if the cell receives the signal to go at G1, it will divide and complete all the other phases normally. If it doesn't receive the signal at G1 to go ahead, then usually it will exit the cell cycle and switch to a non-dividing state known as the G0 phase. So a lot of cells do go to the G0 phase, at least for some point in their lives. Mature nerve and muscle cells never divide, so they're basically permanently in G0. And then other cells can be called back from G0 to divide when necessary. So for example, cells that undergo mitosis, they can be called back from G0 and go through mitosis in order to divide. So this is kind of a visual representation of what happens. If it's like a stop and go signal, so at the G1 checkpoint, if it doesn't have the go-ahead signal, the cell is going to enter G0. But if it does get the go signal, it's going to continue through the cell cycle. And then in a similar way, we have the M checkpoint. And this is usually regulated by, for example, the chromosome attachment to the spindles. If the chromosomes aren't fully attached and lined up, mitosis will not continue past pro-metaphase. So that's what we call the M checkpoint. But if the chromosomes are attached and lined up, that means it's going to divide correctly, and that's kind of the go signal, and then the cell division will proceed all the way through. How this relates to cellular communication is that the cell cycle is actually regulated by proteins and protein complexes. Once again, those proteins communicate with each other. And the 2 different main types of proteins involved are known as protein kinases, which you've heard about already, and proteins called cyclins. One example of an internal signal, which we've talked about, is the signal at the M phase checkpoint. So this is what you saw in the bottom half of that picture. So if the chromosomes are not attached to the spindles properly, anaphase will not begin. However, if those kinetochores are attached appropriately, that activates a regulatory protein complex, which activates an enzyme known as separase. And as you can imagine, ACE, it's an enzyme, and it's separating those chromosomes. And so the sister chromatids separate, and then anaphase can actually begin. So that would be an example of an internal signal from the cell. Some examples of external signals, which are factors that control cell division outside of the cell, are known as growth factors is one example. There's a lot of different types of external cellular control, but growth factors are proteins that are released by certain cells that stimulate other cells to divide. So, for example, there's platelet-derived growth factors known as PDGFs, and they stimulate the division of human fibroblast cells. So once those growth factors are released, that is the signal to those fibroblast cells, oh, we need to divide and make more. Another external signal is known as density-dependent inhibition, which is a really cool phenomenon. Basically, if the cells get too crowded, they'll stop dividing. And so this has to do with the binding of cell surface proteins to their counterpart on an adjoining cell. So it's still controlled by proteins, but it's outside of the cell. It's not within the cell. And then there's, in a similar vein, something known as anchorage dependence, which is another external control of the cell cycle. And in this case, typically if cells want to divide, they have to be attached to a substratum or a base of some sort. So for example, a flask or an extracellular matrix of a tissue. If they aren't attached to anything, they typically have trouble dividing. So one thing that sets cancer cells apart is they do not respond to either of these external controls. They act independently of those things. So if it's crowded, they don't care. If they don't have any anchor, they don't care. Cancer cells don't follow these signals. So this shows you a picture of anchorage dependence. You can see these cells need something to attach to in order to divide, some type of surface. And then this is an example of density-dependent inhibition. So once the cells have filled the single layer, they're not going to divide anymore. It's an external control that causes them to stop dividing. But if we were to remove some of those cells, they would fill the gap until once again they had formed a single layer. Cancer cells don't abide by those external controls, and you can see here they've really overlapped and even formed on top of each other. They don't necessarily require that surface to anchor onto. So our cell cycle controls do not apply to cancer cells, like I mentioned. They actually don't respond to signals that normally regulate the cell cycle. They don't need growth factors to signal them to begin dividing. A lot of times they make their own growth factor, or they don't even need a growth factor. They'll just keep going, or their cell cycle control system may be unlike any of our other cells. And that's part of the reason why they were such a mystery to us for so long. A normal cell is converted to a cancerous cell by a process known as transformation. And when a cell has been transformed, we consider that to be a cell that is able to divide indefinitely in a culture. So these cancer cells typically are able to divide without any other types of controls, and sometimes even deprived of nutrients. And in the case of cancer cells, if they are not eliminated by the immune system, they can form tumors. And these tumors are masses of abnormal cells within otherwise normal tissue. So usually, if your body produces a cancer cell, your immune system will recognize and destroy it. But if not, a tumor will form. If those cells remain at their original site, that tumor is known as a benign tumor, and the cells, that means the cells haven't moved to any other parts of the body. Malignant tumors, on the other hand, are cells whose either genetic and or cellular changes allow them to spread to other tissues. And once a tumor has spread to other tissues, we say that cancer has metastasized or undergone metastasis. And this is when these cancer cells have been sent to other parts of the body. And that's usually much more serious because they might form additional tumors and impair the functions of other organs. This is an example of a breast cancer tumor, and once it's been metastasized, you can see the tumor may have traveled, and usually that happens through either the circulatory system, blood vessels, or the lymph nodes. There's since been several advances in cancer treatment because by understanding how these cancer cells' cell cycles differ than normal cells, we've been able to come up with some more personalized treatments. So cancer cells may have an abnormal number of chromosomes, which may be one reason why they act the way that they do. Others may have their metabolism altered, and those might cause different organs eventually, if they spread, to cease to function. Some of them may have changes to their cellular surface, meaning that they might not have an attachment to the extracellular matrix. Remember, they don't need an anchor necessarily, which allows them to spread to other tissues nearby. And like I said, with metastasis, they can actually separate from wherever they started and enter the blood or lymph vessels and travel to form new tumors. So one way that we can treat cancer is through something known as radiation, and this typically damages the DNA in cancer cells because cancer cells seem to have lost the ability to repair DNA in the S phase once it's been replicated, whereas our normal cells have a lot of checkpoints where they're able to check. And so radiation tends to be a little less difficult on the body just because it's only really targeting the cancer cells. The other treatment that you probably have heard about is known as chemotherapy. And these are when drugs that are toxic to actively dividing cells are administered to the body. However, these drugs interfere with specific steps in the cell cycle, and because it's meant to affect cells that are actively dividing, the side effects of chemotherapy also act on cells that frequently divide in your body. That is why most of the time, if you've known someone who's gone through chemotherapy, they may have lost their hair because hair cells are actively dividing. They get nauseous because your intestinal cells are being affected, and they typically are always actively dividing. So those are two of the main types of treatment that we have for cancer cells.